Good morning and welcome to the epilepsy seminar series at GW. As usual, quick reminders, people in the room, kindly silence your phones. People in the room and on the web, you will get a 5-digit code afterwards to text for CME credit. This morning, I'm delighted to introduce Dr. Kevin Staley. Dr. Staley trained in physics at Loyola Marymount University in Los Angeles and in medicine and pediatric neurology at the University of California, San Diego, in cellular electrophysiology at Stanford. And he is currently the Joseph P. and Rose F. Kennedy Professor of Child Neurology and Mental Retardation at Harvard Medical School and the Chief of the Section of Child Neurology at Mass General. Dr. Staley has been studying neuronal ion transport for 3 decades. He has secured sustained NINDS support, which made possible a bench-to-bedside translation of basic science findings to a successful multi-center clinical trial for ion transport inhibitor bumetanide for neonatal seizures. Today, he will tell us about his latest research and how it uncovered a broader and more complex picture of neuronal ion transport. His new insights will hopefully provide a new translational opportunities for the treatment of acute brain injury and intractable epilepsy. Dr. Staley.
Thanks for the invitation. There's been a revolution in genetic diagnostic power for epilepsy, probably about 50%, so the majority, not the vast majority, but certainly the majority of patients we see with early epileptic encephalopathies now, we can diagnose between the MRI and various genetic studies. And there's a genetic therapeutic revolution now too, with spinal muscular atrophy, a universally fatal disease of infancy is now treatable. And hopefully soon, some of the epileptic encephalopathies will be also, there's already been experimental therapies for neuronal ceroid lipofuscinosis. So there's tremendous excitement in terms of genetic epilepsies, but where are we for acute brain injury? I would argue we haven't got to first base. So we still have a lot of difficulties with the fundamentals of care of patients who are acutely brain injured. And we have problems with cerebral edema, seizures, and chronic epilepsy. We haven't really gotten very far with that. And I'm going to argue today that the reason for that is that we don't understand the basis for significant secondary injuries that cause these problems, and that maybe with looking at this problem from a new perspective, we can get a lot farther therapeutically and diagnostically. So disclosures, I don't have any financial disclosures. These are just NIH trials. I should disclose the people who actually did the work. So the top row and Kyle are all junior faculty now, so this is a project that's been going on for a while. But I'm grateful to everybody, especially Janet Soul, who did the clinical trial of bumetanide for neonatal seizures that I'll talk about in a minute and that Mohammed had mentioned. So the outline today, the challenge of acute brain injury, cerebral edema, and acute seizures. We're going to talk about some new physiological insights about how neurons manage ions in water. It's not really the way we were taught. The derangements that happen after brain injury, then that gives us new insights into these intractable problems of cerebral edema, acute seizures, chronic epilepsy. I doubt we're going to get to the synaptic physiology talk, but we'll see how fast I can talk here. So this is the sort of cliff notes of today's talk. It's a paper we published in Trends in Neurological Sciences, Neurosciences a couple of years ago. And it really gets at this idea that there is a constellation of things that happen with acute brain injury. It doesn't really matter if the injury is infectious or traumatic or ischemic. What we tend to see is diffusion-weighted changes, so restricted diffusion, seizures, mass effect, and more seizures. And so on the left is a patient with neonatal herpes encephalitis, and on the right is an adult with MCA stroke, but very similar problems in both cases, right? Restricted diffusion, mass effect, particularly obviously on the MCA stroke, and seizures. And so the question is, why do these things cluster? Why is that such a common modality or sort of theme in the ICU? Here you see about 17% incidence of epileptiform abnormalities in an early monitoring study, and that number 17% will come up later when we talk about therapies. And obviously the more cortical the problem is, the more likely you are to have seizures associated with that. Neonatal seizures have a variety of etiologies, but really 80% of them are acute injury. And so I focused for much of my career on the treatment of neonatal seizures. The nice thing about this is it's an acute injury and you know exactly when it happened. You know, this is something that happens during parturition, and so you can time that very nicely. So here's a couple of studies of the time course of neonatal seizures. So birth is at the if I'm mouse on, yeah. So birth is here, and so you see that they develop in the first 12 hours, peak about 24 hours, and then dissipate by 4 days of life. These are uncooled babies. If you look at a cooled series from CHOP, it's about the same distribution, maybe a little bit longer tail. So if you can see that gray shaded area. This is 72 hours of cooling and then 2 days of warming up. You can see a little bit longer tail, but pretty much the same distribution. So this acute bolus of seizures that happen really at 24 to 48 hours of life and then taper off. And if you look at the MRI studies, we see that as the seizures are peaking, the MRI diffusion is decreasing, and as the seizures start to taper off, the MRI diffusion restriction improves. And so diffusion restriction means that cells are swollen, there's cytotoxic edema, and there's less extracellular space, which is where the diffusion actually happens that you pick up on the MRI. So that was ischemia. We see similar things with trauma. So if you look at ICP problems with trauma, peaks in 24 hours and tapers off over 5 days, similar to what we saw with acute hypoxic-ischemic injuries in babies, and the seizures have the same time course. We'd like to treat, but we don't really have very good treatments. You know, if I once gave this talk, and a neurosurgeon in the audience raised their hand and said, "We have great treatments for cerebral edema with craniectomy." So I've included these slides from that point on. So this is the DECRA trial, the Australian trial for craniectomy after diffuse closed head injury. And you can see from this graph, the yellow is control and blue is after craniectomy. It does decrease the intracranial pressure, but it didn't improve outcomes. So the pink or brown is the people who did poorly, so they had low Glasgow outcome scores. People in green did better. And so decompressive craniectomy, which is the top plot, so there are more people in the dead or disabled group than in the standard care. So even though you can lower pressure, it didn't help in this particular circumstance. For MCA stroke, malignant MCA stroke, we have a lot of definitions of that. So someone with early symptoms or concerning imaging findings, so a large volume of restricted diffusion or midline shift. We've had a lot of studies to look at craniectomy, and this is a little bit better than trauma, but not much better. So if we look at improvements after craniectomy and MCA, we see that the best outcome, which is a very mild deficit, increases by a factor of 7. That's great. The second best outcome, which is hemiparesis but still ambulatory, increases by a factor of almost 2, which is great. But here's the problem, is that a bad outcome, which is that you're alive but dependent, increased the most by a factor of 15. So this makes for a very difficult conversation with the family if you're going to propose craniectomy for malignant MCA. And as an epilepsy group, if you look at the incidence of seizures after craniectomy, it's pretty spectacular. Remember we said it was about 17% incidence of just epileptiform abnormalities on EEG after acute stroke. After craniectomy, the incidence of acute EEG seizures is about 50 to 60%, and the incidence of chronic epilepsy is far higher, 3 times higher than what we see from other MCA strokes. Now you can argue that this is the most severe stroke and most of these people would have died anyway and maybe you rescued them, but nevertheless these are significantly difficult outcomes to decide whether or not craniectomy is beneficial. If we look at treatment for seizures after brain injury, we have the same problem. So again, I'm a pediatric neurologist and focus a lot on neonatal seizures, but I would say it's a model for any seizures after acute brain injury. So this is just my rough freehand drawing of the incidence of seizures after hypoxic ischemic injury in the neonatal period. I actually drew this long before those studies came out, before long-term monitoring of the neonates was standard practice. But I think this is pretty accurate in terms of the incidence of seizures. This is a study in the bottom of the blue bars from 1999. Remember when this study came out 20 years ago, I was completely underwhelmed. This was a study of phenobarbital versus dilantin for the treatment of neonatal seizures. And even in 1999, those drugs had been out for 60 or more years. So I thought, well, what's so innovative about this trial? But what was innovative about it was really the design of the trial. So this was the first neonatal seizure trial to take the natural history into account. So they asked, were seizures increasing during the baseline period of 2 hours before treatment? Was it stable or was it decreasing? And what you can see when you look at the blue bars is that there's a really powerful effect of the natural history on the response to drug. And if you read about this study in a textbook or review, they'll say, oh yeah, about 60% response to dilantin or phenobarbital. But that's obviously weighted by these guys here. Anything's going to respond here, right? You could give saline and get a great response at that point in the natural history of neonatal seizure. So it's really only this first bar of 25% response rate when seizures are increasing. That's where you'd really want to see an effect, and that's where it's most lacking. Just to emphasize the natural history, if you look at a study where they don't think about the natural history, here's a trial of Keppra, pretty benign anticonvulsant. We all feel comfortable giving that IV. And if you look at this study, they said, okay, well, seizures started, in the first 12 hours, and then there was an average of, I think, 36 hours of fiddling around with phenobarbital and dilantin before they got the study drug. This is an open label uncontrolled trial. And this pink is where they got Keppra, and you say, 32 response in an hour, 64% 24 hours, 86% 48 hours. You say, shoot, I'm switching to Keppra. This drug looks great. But this is the natural history of neonatal seizures. So this is the uncooled study that I showed you before from Ireland, and you can see that you could have given saline at these points and gotten the same response. So this is just the natural history of neonatal seizures, and so without a control group, you really can fool yourself. And I spent an hour at this poster trying to convince this group that maybe they should have had a control, and at the very end, the pharmacist who had run the trial said, well, are you going to use Keppra now? No. Anyways, but it's hard. So if you don't look at it this way, it can be difficult. So we have this problem. We have acute brain injury. We all know the problems in the ICUs, neonatal, pediatric, adult ICUs of acutely brain injured patients. And so I'm going to tell you about one brick in the wall of neuroscience knowledge. It may seem to be a very small and unimportant brick, which is how neurons handle ions, particularly chloride. And in the 90s, we realized that chloride, we used to think, oh, it's just passively distributed. It's like totally irrelevant. The distribution of chloride depends on the distribution of everything else. But in the 90s, we decided, no, chloride is actually actively transported. We identified the transporters that move chloride in and out of cells. And I was excited at that point, because I was working on chloride transport back then. And I really struggled to find clinical relevance for these new discoveries. And the bumetanide trial I'm going to talk about in a little bit was an outgrowth of that, but it was difficult to translate it. About 5 years ago, we published a paper in Science where we said, it's not really active transport, is not as important as we think. Chloride's really displaced by all the other anions in the cell. And once we made that step, a lot of things became much more obvious. And I want to share those things with you today. And this is what we're going to talk about in the next few slides. And then we're going to translate it to a bunch of different things. This is a clinical talk, so we're going to stay on the top row. So acute neonatal seizures, cytotoxic edema after brain injury, and intractable epilepsy. And if we get to all of them, great. If we don't get to some of them, that's okay too. Okay, so in 2005, about the heyday of cation chloride co-transport, the hypothesis that this is what set the GABA reversal potential. It's a beautiful story. There was this transporter called KCC2, but it was a potassium and chloride co-transporter. So it used the energy in the potassium gradient to co-transport chloride out of the cell. So there was less chloride in the cell than what you'd expect from passive distribution. What that meant is if you opened a GABA or a glycine channel, a neurotransmitter-operated channel that was only permeable to chloride, chloride would flow into the cell, and that accumulation of negative charge, because chloride's an anion, would hyperpolarize the cell and prevent action potential generation, and so GABA was inhibitory. And the inhibitory effect of GABA was really predicated on the action of this potassium chloride cotransporter KCC2. And so this is a simple system, and it makes some really, really robust biophysical predictions. One is that the chloride reversal potential has to be the same as the potassium reversal potential, or the transporter is going to keep going until it is the same. It's a very high velocity transporter. The other thing is there's a lot of potassium on the inside of the cell, a lot of chloride on the outside of the cell. So the Nernst equation, it depends on the ratio of these two ions. It's shown here in this middle equation. And so another very robust prediction is that the intracellular chloride is pretty much going to be locked to the extracellular potassium. There should be very little variance in intracellular chloride. And again, this top thing where the chloride reversal and potassium reversal should be the same. The developing nervous system is different. There's a different transporter, so KCC2 isn't expressed. There's this other one called NKCC1, and it uses the energy in the sodium gradient to actually bring chloride into the cell. And so now the idea is when you open the same channel, GABA or glycine channels, there's an excess of chloride inside the cell. When you open the channel, this negatively charged ion is going to flow out. It's going to depolarize it and excite the cell. So GABA is excitatory in the neonate and inhibitory in the older, more mature nervous system. And this NKCC1 has predictions also. The chloride should be very high inside the cell, and it's blocked by bumetanide. So this was sort of the genesis of our bumetanide trial. So why would we care about this chloride concentration? Well, as I said, it sort of determines the effect of GABA. So here's an experiment that we do frequently. We record from this pyramidal cell with a recording pipette, and we stimulate an interneuron to release GABA, and we look at that opens the GABA channel, and we look at the currents. And so if we hold the membrane potential at a negative, minus 75, where's that mouse? There it is. We see that that forces chloride out of the cell, and so that's an inward current. The loss of chloride is depolarizing. If we hold it at a more positive potential, say minus 55, chloride actually flows in, and that's inhibitory. And right at minus 65, nothing happens. And so we can plot these results and find the reversal potential for chloride. If we change this experiment a little bit, so we're not looking at the currents anymore, we're looking at the membrane potential, and we load this, we put a lot of chloride inside the pipette, so we flood the cell with chloride. Now look what happens. When we stimulate the GABA receptor, instead of pushing the membrane potential to minus 65, the reversal potential, we actually depolarize the cell because chloride's leaving, it's negatively charged, and we trigger an action potential. And the point is that under these conditions, when GABA is excitatory, none of our anticonvulsants are going to work. So if we give an anesthetic concentration of pentobarbital, that opens the GABA channel longer for each GABA molecule it binds. And all we do is trigger more action potential. So anticonvulsants are not going to work if we have a deranged chloride gradient across the membrane. And the point then is that there's a whole range of chloride GABA effects depending on what the GABA reversal potential is. So it can go from hyperpolarizing inhibition, the sort of thing you learn about in medical school, to shunting inhibition, where you open the channel and there's no change in the membrane potential, but it short circuits any excitatory effects, to frankly excitatory effects. So you can remove the magnesium block of the NMDA receptor, you can activate low threshold calcium channels or even high threshold calcium channels depending on what the reversal potential is. So the exact amount of chloride inside the cell is very important. So we showed in the 2000s that if we added an NKCC1 inhibitor, so we prevented the accumulation of chloride, we could greatly increase the anticonvulsant effect of phenobarbital. So these were done in vitro. There was no blood-brain barrier. And if you look at that final bar on the left-hand side, you see that phenobarbital plus bumetanide has the most powerful effect on seizures. And on that basis, we started a neonatal seizure trial. This started about 2009. We got funding. So neonates who were at risk for seizures got continuous EEG monitoring, which in 2009 was sort of an innovation. It's not the standard of care now. We saw seizures, electrographic seizures, so we pretty much ignored clinical seizures. They were treated with high-dose phenobarbital, 30 per kilo. If they had further seizures, which put them at very high risk for morbidity and mortality, we randomized them to either standard of care, which is more phenobarbital, or standard of care plus bumetanide. So there wasn't anybody who got bumetanide or placebo. You got more phenobarbital with bumetanide or more phenobarbital with saline. So this really was looking at how does bumetanide enhance the effect of phenobarbital more than how does bumetanide work by itself. But this was a much easier thing to convince parents to try. So that trial took 10 years. We just submitted that paper. And I'll give you the results in a little bit. But what did I do in the meantime? I was the basic science guy. And so it occurred to me that these simple predictions could be tested in the literature. I tried to prove this for most of the 90s, and I really had trouble. I always thought that I wasn't doing the experiment the right way. But it's very difficult to show this very sort of monolithic value for E. chloride. I kept getting all sorts of values, and I assumed that there was experimental noise. But finally, I looked at 20 different studies that had used a very non-invasive way to measure E-GABA. So they, instead of patching onto the cell directly, they used an antibiotic, gramicidin, to drill little holes. They're only permeable to cations in the membrane, and that avoided disrupting the chloride concentration. When we looked at these 20 studies, a couple of things were obvious. One was that there was a huge range in the values, a 25 millivolt range in the value for E-GABA. These aren't individual cells. These are group mean values from different labs doing the exact same thing in the exact same animal, exact same gestational age. And if you looked at the potassium reversal potential, it was nowhere near the chloride reversal potential. So this fundamental prediction was not true, and this variance in the GABA reversal potential was something else that wasn't predicted from this transporter. So there had to be something else going on. And so when I moved to Boston, one of the things we did was avail ourselves with all the fancy 2-photon microscopes that were available, and also this great chloride indicator. It's a transgenic, sort of chloride-sensitive fluorophore called chromelein. So cyan fluorescent protein is not sensitive to chloride. But yellow fluorescent protein, the fluorescence is quenched by chloride. So you can look at the ratio of cyan to yellow fluorescence and get a really nice indication of the chloride concentration that is independent of the concentration of the fluorophore. So very accurate. This is pH sensitive, but as long as you do an experiment where the pH stays the same, it's not a problem. And what we found is that the chloride concentration was continuously distributed. So this lower panel here shows the pseudo-colored by that color bar to show the different concentrations of chloride in neighboring cells. And there was a big difference from cell to cell. For me, this was an eye-opener because I had spent a decade patching onto one cell per day or maybe 2 cells a day and getting different values and assuming that it was noise or something else. But it wasn't noise. It was just that's the way it was. The difficulty with this experiment is that that pink and blue arrow show up at the upper right-hand corner show where KCC2 should drive chloride or where NKCC1 should drive chloride. And none of the cells that we recorded had chloride concentrations that were consistent with the action of those transporters. So those are the equilibrium points for the two transporters, and all we found were points in between. So we published this, and you can imagine that everybody who was making a living off of studying KCC2 and NKCC1 was not very happy with us and assumed that we had screwed this experiment up somehow. And so I heard about that for about 5 years. And it was so really nice in 2017, my most vocal critic published the exact same result. So they used a different transgenic fluorophore called clofensor, which can be corrected for pH, and got exactly the same result. So very wide distribution of chloride, none of which could be explained by NKCC1 or KCC2 action. And people had published this with gramicidin perforated patch before, but the numbers were so small it was hard to really put much faith in that. But now we clearly had evidence that chloride was widely distributed. Now you could argue, well, maybe it's widely distributed because KCC2 is pushing one way and NKCC1 is pushing the other way, and so we get this distribution in between. And so that's an easy thing to test. If KCC2 is trying to push chloride out of the cell and NKCC1 is trying to push it in, we could cut the legs out from this guy or that guy pharmacologically, and that should make a big difference in the chloride distribution. So we have specific antagonists for the two transporters, but when we put them on, we saw very little effect. So this was the initial chloride on the x-axis, and the y-axis is the final chloride. And you can see that we don't see the kind of effect we would expect, which is if we block both transporters, the chloride should be distributed passively, and if we blocked one or the other, there should be a radical swing in the distribution of chloride. That didn't happen. It didn't happen in neonates, which presumably were NKCC1 dependent, or in adult animals, which were KCC2 dependent. Same experiment. And again, the people who didn't really believe this replicated this experiment exactly and got the same result. So again, they couldn't get the transporter and antagonist to change the chloride distribution either. So what is setting the chloride reversal potential? Well, when I did those experiments in the 90s, if I wanted to change the chloride concentration inside a cell, I'd put a different concentration of chloride in the pipette and add some impermeant anion like gluconate or methyl sulfonate. And then you'd rupture the patch and then measure the chloride reversal potential. And the idea was that this stuff would flood the cell. The cell was like a big water balloon waiting to be filled with this dialyzed by this electrode solution, and you'd get the reversal potential. And this works very well for low chloride. And when we tried this experiment with high chloride pipettes, it didn't work so great. And the reason is, of course, there's all kinds of other stuff inside the cell that is not going to be dialyzed by the electrode solution. So the cell is full of, actin and tubulin, nucleic acids. Those are all anions, and they're all polymers, and they're not going to float out of the cell. Chloride is a very minor constituent of the ionic milieu of a cell. It's about 5 or 10 millimolar, maybe 20, but there's 150 millimolar of anions in there, most of which are amino acids that are negatively charged at physiological pH, like glutamate, and things like tubulin, which are part of the cytoskeleton, have long barnacle-like polymers of glutamate hanging off of them. So there's a lot of negative charge accumulated inside a cell, and actin is also very negatively charged. So if you do this experiment, you can actually show that the more chloride you put into the electrode on the x-axis, you have a bigger and bigger error when you try to dialyze the cell. So the cell is resistant to dialysis because of all these immobile anions. So that's great. It certainly explains why we get so much variance from cell to cell in terms of the chloride, because as I'll show you in a minute, the concentration of these proteins changes from cell to cell and from different parts of the cell. But this sets up a very difficult problem that's directly relevant to cerebral edema and acute seizures after brain injury, and that is, this is what you will see if you look in your old Bio 101 textbook for a Donin equilibrium problem. So you have permeant ions, potassium and chloride that can go through the transporter. Water can go through the transporter also. And you have impermeant anions, so things that can't go through. And this sets up, the solution for this ends up with an osmotic gradient where water wants to get in. And just to make this a little bit more intuitive, this is the same thing that you try to do when you boil brats in beer. The idea is that this is going to make the brats nice and plump and juicy. They're going to swell up, but that is not what we want to happen in the neuron. So if we just think about the brat situation for a second, we have brat, we have beer. Water is supposed to go inside the brat when you incubate it. Now why does that happen? It's not from the sodium chloride, right? Because the sausage casing is permeable to salt. What really causes the water to go in there is all that mystery meat inside the brat. So ground up proteins in there are trying to get wet. Water wants to wet those things and reduce the concentration of the proteins inside the brat to the concentration on the outside, which is 0. So basically that brat is going to swell indefinitely if you leave it in the water long enough. We don't want that to happen to the neuron, right? So how does a neuron keep from ending up like a swollen brat? It's the same biophysical situation. And so the solution is the same as a cliche as old as life itself. Bacteria have a way harder osmotic challenge than neurons do. Neurons sit behind the skin. They have kidneys to regulate the osmotic tension. Bacteria don't have any of that, right? They're sitting in your gut, and they're happy at 300 millimolar, so everything's isotonic. But at a moment's notice, they could end up in pure water. They could get flushed out in the ocean. It's 1,000 milliosmoles, so they can have these dramatic changes in the tonicity of their surroundings. And you can imagine a bacterium in a muddy puddle where that's at 300 millimolar, and then it starts to rain, it's going to go to 0 millimolar. So how does that bacteria prevent osmotic rupture under those circumstances? And the answer is the same as for the M&M, right? M&M has a carbohydrate shell that protects it from the environment, like your kid's warm, sweaty hand, keeps it from melting. And bacteria have these, basically a carbohydrate shell also. It's peptidoglycans, so it's a combination of proteins which anchor it to the membrane, and then long strings of carbohydrates. And the point is not to make a physical girdle, which is the way I learned about it in medical school. The point is really to retard the diffusion of water through that columnar structure of the peptidoglycans. Just like the long neck on a wine bottle. If you ever left a wine bottle in the fridge without a cork, you can leave it in there for a month, and you're not going to have a change in the level of wine, because the long neck retards the diffusion of water out of the evaporation of water out of the wine bottle. That's why they make them that way. And so probably the M&M is not the right analogy for this. Probably a little puppy here who would freeze to death in a couple of minutes if he didn't have any fur, right? He would just transfer all of his heat to the snow and he'd die of hypothermia. Puppy's happy as a clam, though, because he has this fur which greatly retards the diffusion of heat out of him and into the snow. And so neurons have the same kind of fur. You've all heard of these things probably called perineuronal nets. They make these beautiful pictures. They're most obvious around these parvalbumin positive basket cells, so inhibitory interneurons. But the only reason that's why we hear about them in relation to interneurons is because that particular motif of carbohydrate shell is recognized by Wisteria agglutinin, which is sort of a plant antibody, and it recognizes a particular sequence of starches on these things. But if you look back in the 1890s, Golgi had beautiful pictures of neurons with perineuronal nets that weren't all interneurons. Every neuron essentially has these things when you use an ionic stain. And we published this also. So Golgi used an iron stain in 1890, and then we had the temerity to publish this 100 years later, but we used a copper stain. So that was okay. But anyways, these things, basically the fur are these long strings of glycosaminoglycans. It's like a sugar, so it's a long repeating chain of sugars. They're not called starches because they have an amine group on them, so they're glycosaminoglycan, a lot harder to say, but basically a long starch. Long 300 stretches of starches, very, very heavily sulfated. So you can have a -9 charge from this particular dimer. And it is, these are the most heavily charged biomolecules that we know of. So they can not only retard the diffusion of water, they can just have a significant osmotic impact and serve as a balancing Donin force for your intracellular things like actin and tubulin, and they can also displace extracellular chloride. So we'll look at all those things in a second. What I'm trying to say here is that chloride is transported. That certainly happens, and after you've had a synaptic current, you've moved chloride one way or the other, the transporters serve to get things back to equilibrium, so they're critical. But the actual value of chloride is due to displacement by all these other macromolecules. So actin and tubulin on the inside, and glycosaminoglycans, sulfated glycosaminoglycans on the outside. And so when you think about it that way, then it makes perfect sense that the chloride reversal potential is not the same as the potassium reversal potential. Because if we have a situation like this, what's left out of this cartoon, which I drew in 2003, I didn't draw it myself, but I gave the artist the idea, was that we left out all the stuff that's inside the cell. So this is not a vacuum up here. It's not an air bubble inside a neuron. It's chock full of other stuff, these anionic polymers. And each neuron has a different amount. And these things are very highly charged. So they actually have higher charge. Tubulin has a higher charge per angstrom than chloride does. So if you look at, now we've got these beautiful ways to look at protein distributions inside cell and compare them cell to cell. And you can see that every cell has a different amount of tubulin, a different amount of actin. These aren't actually neurons, but you get the general idea that the amount of any particular protein varies from cell to cell and also varies within a cell. So this is a single neuron, I forget what it is, some kind of fibroblast. But you can see these are all the different protein species that they had antibodies to, and what they did is a very laborious experiment. They washed on one antibody and then wash it off and put on the next antibody and wash that one off. They did it 41 times, and they got this distribution of all these different proteins. But you can imagine if you have all these different proteins with different negative charges per protein, that you're going to have a wide distribution of chloride also in different parts of this cell to balance the local charge. And on the outside then you're going to have these glycosaminoglycans, this sort of fur on the outside of the membrane to balance all that dominant force generated by those proteins. So just a couple of data slides before we get to the clinical stuff. So this is X-ray fluorescence microscopy essentially. So if you use a high enough short enough wavelength light, elements are fluorescent, and you can measure their fluorescence. And so these guys in Germany did that, and they saw the elemental profiles of different neurons. So these are pyramidal cells, and they scanned the beam across this pyramidal cell up here between those two stars. So they went from the extracellular space to the cytoplasm nucleus into the extracellular space on the other side. And what they saw was what we had expected, which is, so if they used the wisteria agglutinin, which was labeled with nickel, they saw a lot of glycosaminoglycans. So this is in millimolar, so 80 millimolar of this stuff. They saw a lot of sulfate. Again, these glycosaminoglycans are polysulfated, so very high concentrations of sulfate in the extracellular space, and high concentrations of phosphates, which I think are green, in the intracellular space. So lots and lots of things. Now the one weird thing about this paper is that they were never, these were fixed cells, so exactly how to translate millimolar fixed tissue into millimolar of live tissue wasn't really clear. It's probably why it ended up in scientific reports. But nevertheless, you can see that there are very high concentrations of these non-chloride ions in the outside of the cell and on the inside of the cell. And so we've done the same thing. So this is a paper we've just submitted for publication. We looked at chloride microdomains inside the cell. And so this is, I showed you those 41 protein distributions a couple of slides ago. And so this is sort of the negative image of this. You think about a photographic image. So wherever there's a lot of proteins, there's going to be little chloride. And wherever there's not very much protein, there's going to be high concentrations of chloride. So this is an interneuron in the hilus of the hippocampus from a mouse. And reversal potential and the effects of GABA in those situations. Here's a pyramidal cell, same thing. We zoomed in on the dendrite, and these are different chloride concentrations. We're limited to submicron resolution. We can't get down, obviously, to atomic or molecular resolution, but pretty good resolution. And you can see these microdomains. And these are just the distribution pixel by pixel of the chloride concentrations in the interneuron and the pyramidal cell. This is just to say that it's not noise. So you might say, well, that's great, but maybe if you took another photomicrograph a second later, you'd see something completely different. Maybe you're just looking at the noise on a TV screen and fooling yourself. So we did a lot of stability experiments. I'm not going to show that shows that's not the case. We did transport inhibition experiments that show this is not something that depends on transport. And this is just to say it's not dependent on Brownian motion or white noise either. It doesn't have the right spatial distribution for either of those things. So an interesting thing then is if you have these variations in chloride, does it affect GABA signaling? And there's been a lot of, probably 20 papers in the literature over the last 20 years saying that there are subcellular differences in the chloride reversal potentials. This was the first paper that really made a big deal of it. It was Sabatics et al. Science 2006. And they found that when they stimulated basket cell synapses, these are GABAergic synapses, that landed on the soma, they got a reversal potential minus 77. When they did axonic cells, they got a reversal potential that was 20 millivolts higher, minus 54. So they did it by interneuron cell type, and they thought there was something about the interneuron cells. But we did the same experiment and just did a bunch of basket cells. So these are basket cells that express green fluorescent protein. We patched this pyramidal cell in the middle with this dark pipette and filled it with Alexa so we could see where it was. All these light cells are interneurons that express green fluorescent protein and are driven by parvalbumin. And what we can see, and then these are the reversal potentials. So we puffed glutamate on them and excited the interneuron to release GABA onto the principal cell. And each of those synapses reversed at a different place. So this cell had a 12 millivolt shift across the different synapses, different reversal potentials. So those are going to have much different effects on the cell. And here's all the experiments that we did. This is the range of interneuron stimulations in red. So it goes across 25 millivolts effects. And this, just for comparison, is that range of group mean E-GABAs that people got from gramacide experiments looking at the reverse potential of GABA. So there's a very wide range. So these microdomains that we see using these fluorescent dyes really seem like they have a direct impact on GABA signaling. And we can go more into that if we have time, but we probably are not going to have time. Let me focus on the clinical implications of this idea that GABA is displaced by charged macromolecules. And I'm going to give you the Reader's Digest version of the cerebral edema part. We've done a ton of experiments to examine every single step in this pathway. I don't have time to go over all those. But the idea is, you think back to our experiment of the M&M or the fuzzy dog. What happens if you take off the sugar coating or you shave the dog? Or for bacteria, what happens if you disrupt the bacterial wall, right? So we've all heard about this, that's how penicillin works. You disrupt the bacterial wall and it's like taking the girdle off, the cell just pops. Well, that's not really what happens. You don't, first of all, you don't dissolve the peptidoglycan wall, you just inhibit its synthesis, and the cell outgrows its wall, and then it dies an osmotic death the first time it has an osmotic challenge. Because it just, it has a lot of proteins on it. Bacteria have a lot of negatively charged proteins too. There's a Donin effect, and they swell and they pop. So that's how penicillin works. And unfortunately for us, neurons have a very similar thing. So a healthy neuron has an extracellular matrix around it. These are all the glycosaminoglycans that we saw pictorially before, sulfated glycosaminoglycans. When a cell dies, it has to remove that glycosaminoglycan shell. And so cells have metalloproteinases, matrix metalloproteinases, or MMPs, that they release when they die. And when they necrose, they release those in an uncontrolled way. And if you have a stroke and you have billions of neurons die, you release massive amounts of matrix metalloproteinases, which is not good. So those things dissolve the matrix of the healthy bystander neurons. So these would be the sort of penumbra. You dissolve the matrix of the penumbra, you're going to release glycosaminoglycans to circulation, so you can measure this in the CSF and potentially in the blood. We have a Department of Defense grant to look at that. And then that matrix is going to be replaced by chloride. So our friend NKCC1, which exists on the membrane of this healthy neighbor, also exists at the blood-brain barrier and is responsible for transporting salt into the brain, potassium chloride. So chloride is going to replace the lost glycosaminoglycans. So now you're going to have an extracellular matrix with very high concentrations of chloride compared to the control situation. And then the transporters in the brain are going to equilibrate. So if you have higher chloride on the outside, these are equilibrative transporters. They're going to pump salt on the inside, and then the neighbor is going to become unhealthy. It's going to swell. And that is going to restrict the extracellular space, and that's where our MRI diffusion signal comes from. This swollen cell is still synaptically connected, so this guy's not connected anymore. This guy's still synaptically connected, but now his chloride concentration is higher than it was, and so GABA is much less effective in that cell than it used to be. And so this is the basis for the restricted diffusion, the restriction of the extracellular space. The high chloride is the basis for the acute seizure. So you can see that as a consequence of, you can see our hypothesis that we're getting ready to publish is that the damage to the extracellular matrix is responsible for lots of the secondary problems that we have in, after acute brain injury. And so this is sort of the overall scheme. Injury to the primary cells is going to be obviously some cells that die immediately in brain injury. Release metalloproteinases that release these glycosaminoglycans. NKCC1 at the blood-brain barrier transports chloride into the extracellular space and increases the extracellular chloride, that's CLO there. And then these transporters are responsible for bringing potassium chloride inside the cell, which causes it to swell. Because again, the cell can't get rid of its actin, can't get rid of its tubulin that fast. So it's going to swell if you put potassium chloride or sodium chloride inside. So I'm just going to restrict. So we've looked at every step in this pathway, and we have all those results if you're interested afterwards. But I'm just going to focus on two therapeutic elements here, the inhibition of metalloproteinases and inhibition of NKCC1 and KCC2. So a very quick way to look at secondary injury is to use a 2-photon microscope and to use the laser to focus the laser power on a single cell and boil the cytoplasm, and that cell pops. And you can see that here. Here's the damaged cell. And around that cell, there's an increase in the chloride in the neighbors. And that happens from dissolution in the matrix and that increase in potassium chloride that I just explained. Now, the other thing that happens to these once healthy neighbors is that they activate caspase. So that swelling causes a lot of those cells to undergo apoptosis. And pink is FLICA is a fluorescent inhibitor of caspase activation. And so it's an easy marker for caspase. So that's in control. You can do lots of experiments where you lower the laser power slightly so you don't pop that cell and you don't get any change in the neighbors. So this is not heating of the neighbors or anything else. This is damage to the neighbors caused by the rupture. If you do the same experiment in low chloride solution, there's no change in the chloride concentration. So clomillion doesn't report any change in chloride in the neighbors, and you have a lot less FLICA activation also, a lot less caspase activation, damage to the surrounding cells. Now, we thought that furosemide ought to work like a charm. This blocks both NKCC1 and KCC2, but it had pretty modest effects. And we had seen that before in the slice model of trauma. So when we look to see, when we make slices and traumatize neurons by the razor blade, we look to see how well bumetanide inhibition of NKCC1 prevented chloride uptake. It prevented it a little bit, but not as well as we had wanted it to. So transport inhibition, there's something about this step at the end there with that second question mark that isn't quite as effective as we had hoped. And we saw the same thing with oxygen glucose deprivation last year. This is in the electronic annals. But what worked great is when we inhibited metalloproteinases with a zinc chelator. So all the metalloproteinases, there's a huge family of these, 50 of them. They are all zinc dependent, and if you chelate zinc, none of them work. And you can see that this works just as well as low chloride. So remarkable preservation of the extracellular matrix. There's no chloride accumulation. The only cells that activate caspase are the ones that were radiated with the 2-photon laser. So metalloproteinase inhibition looks really promising. This is the dose response. Now, you can't chelate zinc without causing all kinds of other problems. So we used a more specific inhibitor of proteinases. So this is back in the slice model. Again, when you make brain slices, when I was a physiologist, I thought that was a perfectly fine thing to do, but you can also think of that as a diffuse brain injury from the razor blade. And that causes a lot of high chloride cells where some cells have been ruptured and they release metalloproteinases, and that problem spreads throughout the slice. So you can have very high chlorides. And much less chloride when we inhibited these metalloproteinases specifically. So MMP2 and 9 are the ones that are most commonly released in neurons. And so there are specific enzyme inhibitors that don't involve chelation of zinc. And you can see those save the cells. It's probably best shown here. So if we look at 0 to 50 microns from the cut surface, we see a modest effect of MMP inhibition, but that's because there's a lot of direct damage there. We wouldn't expect a very big effect. But if we look in the penumbra, so 50 microns below the cut to 100 microns below the cut, we get a really robust salvage. And then down here, the salvage becomes less because there's less damage. So basically, a very nice demonstration of penumbral salvage by these MMP inhibitors. Now, NKCC1, I think, is important in the blood-brain barrier. There is a lot of NKCC1 at the blood-brain barrier. This is a photomicrograph of the basement membrane and the pericytes and astrocytes, and there's little gold particles wherever there's NKCC1. And we find that NKCC1 works great for outcomes. It works for stroke. It works for trauma, to reduce edema, improve outcome. It inhibits seizures when the arteries are open. We had done some of those experiments. It improves outcome after neonatal HI, but there are experiments where bumetanide doesn't work. And these experiments all involve either blockade of the arteries. So this is Shilpa Kadam is a former collaborator of ours, but she did an experiment where she just ligated the mouse pup artery, and it's hard to unligate that. You know, the arteries are tiny, so she just did that with thread. And she found that as long as the artery was blocked, bumetanide didn't work. And similarly, if you damage the blood-brain barrier with collagenase, where NKCC1 is, then bumetanide doesn't work in that situation either. So it does seem like bumetanide, the locus effect is not on the neurons, but probably on the blood-brain barrier. And so if we think about ameliorating matrix damage after brain injury, we look at these treatment points. This is a modestly effective treatment point, but there are, you can plug up some of these salt water fluxes at the membrane, but not all of them. There are a lot of pannexins and a lot of other transporters in the membrane that probably serve to cause net sodium chloride and water influx. So that's not as effective as we wanted it to be, but bumetanide inhibition at the blood-brain barrier seems pretty effective, or effective enough to use it today. MMP inhibition is the future. There are a lot, you've seen all these drugs used before in different situations. We have tons and tons of pharmacological candidates because they were used for cancer therapy for a while but failed because of side effects. And so there are multiple side effects. We don't know all of the effects of metalloproteinase inhibitors, but this is clearly the future over here. I'm pretty excited about that. So again, we have a TINS paper if you want to read up more about that. Let me talk a little bit about our neonatal seizure trial before we quit. We started in 2009, and in 2015, the European group had done a faster trial, so they used a Bayesian trial design, no control group. They wanted to show that bumetanide worked, and they wanted to do it faster than we could. But they ran into some toxicity, so some patients had hearing loss. And so they decided to stop the trial, and they concluded that bumetanide probably doesn't improve seizure control and might cause hearing loss, so it's a terrible drug. And this is in the middle of our trial, which wasn't what we wanted to hear. But then these guys in Norway said, well, wait a minute, you only had 14 patients, and 5 of the 14 didn't have seizures in the baseline period, but you included those guys anyway. So then these guys wrote back and said, no, no, no. We firmly disagree with the conclusion that bumetanide might work, and we think further they disagree with the idea that further clinical studies of bumetanide are warranted. You know, we're in the middle of a trial, so this is not what we want to see in Nature reviews. But they also published, these are the actual 14 patients. And so you can see there are patients like this, patient number 1 who didn't have any seizures during the baseline period, and so he was counted as a bumetanide failure, because you can't have any less seizures than none. And they had a 2-hour baseline period, so this guy, this guy, this guy. There were 5 guys in there that they just counted as failures because they didn't have any seizures in the baseline period, which I would argue is not really fair, right? You can't, those guys can't possibly have an effect. And if you look at, if you just stand back and look, you can see there's a lot more blue on this side of the dotted line than on this side of the dotted line. And this is where they gave bumetanide. But they also gave a lot of other stuff. They gave midazolam, a lot of phenytoin. And so it's really difficult to know what's going on. If you do the statistics, this is a significant effect. But without controls, you don't know, is this natural history? Is this decrease the effect of other anticonvulsants? And is the toxicity due to the bumetanide, or is it due to something else? Janet Soule has soldiered on for 10 years doing this trial. I initially wanted to do it myself, just out of pure egomania, but I decided it was going to be too much work, and so I let Janet do it, and I'm really glad that I did, because it is a huge amount of work to do a clinical trial. So again, continuous EEG monitoring, we looked just at electrographic seizures, we gave them phenobarbital, and then either more phenobarbital or phenobarbital plus different amounts of bumetanide. And here's the results. So this was double-blind, randomized, placebo-controlled, hadn't been done this way in neonatal seizures before, and we were going to do 40 patients. NIH kept telling me, you know, this is a phase 2 trial, it's just not an efficacy trial. My feeling was, you know, I've been doing rats my whole life. I'd never done an experiment with 40 rats before. I was like, that's a huge N. You know, how could this not work? Well, this is how it could not work. So when we randomized, it turned out that the blue dots are placebo. So 6 of 8 of the least severe seizures were in the placebo group, and 12 out of 13 of the most severe seizures were in the bumetanide group. And the reason that's a problem is this is the effect of seizure frequency on response to meds. So this is from that Painter phenobarb versus dilantin study that I told you about in 1999. Mild seizures respond great to phenobarbital or dilantin, and severe seizures don't respond at all. So we're like, oh shoot, all of our study drug ended up in the severe category, and nobody ended up in the mild category. So we analyzed the data a little bit differently. We analyzed it by seizure burden. And you can kind of see here, so this is seizure burden, this is control, this is 0.1 per kilo, 0.2 per kilo, and 0.3 per kilo. This is the only dose that had been tested in people before. And so this is the change from baseline to after treatment. And so you can see a profound decrease in seizures in the most severely affected patients, and that's plotted in individual patients here. So this is total seizure burden, so total amount of seizure minutes, and this is the change in seizure burden. And so what you can see, this is no effect. So this is either you had no change or you had more seizures. And so and this is where you had decreased seizures. And so you see lots of red dots down here because this is all those more severely affected people. But all the people who got bumetanide had a much stronger reduction in seizures, whereas people who got just phenobarbital and more phenobarbital really didn't have much of an effect. Now the other thing you could see here is, wouldn't it be great if there were more blue triangles up here, or blue triangles somewhere over that way, so we could test this hypothesis better, and wouldn't it be better? And I think we have enough red circles here, so we have enough bumetanide. We just didn't get very many patients randomized, severe patients randomized to phenobarb. We also didn't get to the top of the dose response curve, so if you plot dose response, you can see that we should have used more bumetanide, which we were afraid to do because of the hearing problems. So we applied for a drug, we applied for a grant to look at bumetanide toxicity because the NEMO trial had made such a big deal out of it. And it went to an audiology study section, and they didn't give us the money, but in the review they said, well, why don't you look deeper into the audiology literature? They gave a bunch of references, and sure enough, bumetine's already been tried in rodents, which is what we were going to do, and it's not toxic. It's only toxic if you combine it with aminoglycosides. And in the NEMO trial, almost all the patients had been treated with aminoglycosides, and in our trial, almost nobody was, only half the patients, or a third of the patients were treated with aminoglycosides. So we had a much lower incidence of hearing loss, which was probably due to the hypoxemic injury and aminoglycosides. Okay, so I'm going to stop there. Let me just give you the conclusions. So after brain injury, damage to the matrix, I think, contributes to secondary injury. It's manifest as edema and seizures. We didn't get to chronic gliosis, but I think that's one of the reasons why post-traumatic epilepsy is so difficult to treat. You've changed the matrix and the chloride gradient. And so an ounce of prevention with metalloproteinase inhibitors is likely to be worth a pound of cure, but we still have a ways to go. But at least we have a direction now. And I'm excited that we now have things that really work in the lab, and we should be able to translate to the clinic. But as the bumetanide story tells you, one thing I learned is clinical trials take a long time. In 10 years of basic science, the story can change a lot. And so sometimes the story changes in a way that would seem super cool and sexy in 2008 or 9, not so cool later. So you want to be very careful, particularly about these metalloproteinase inhibitors, because again, the reason they failed in cancer is not because they were not efficacious, it's because of all the side effects. So we need to be careful. Okay, thanks.
Thank you so much. I have maybe a long question. You discussed very beautifully the role of osmolarity and the role of the chloride, obviously. But can you put it in a larger context of the complex processes that happen with excitotoxic injury or other insults, including the apoptotic necrotic continuum, the activation of intracellular proteases, immediate early genes? Right.
Good point. So I think that let's see, I guess I don't have a way to, I was going to go to slide viewer. But so in that experiment where we used the laser to pop a single cell and necrose that cell, it's a beautiful model of the penumbra, because everything that happens outside of that one pop cell is penumbral injury. And so we've used it to address exactly the questions you're asking. What about, we've heard for decades about excitotoxicity. And that happens, but it's very brief. So, the glutamate concentration inside the cytoplasm is 2 or 3 millimolar. It's pretty high. And so if you pop a cell from necrosis, or in this case photolysis, you have a lot of glutamate released into the extracellular space, and you only need a single micromole of glutamate to activate glutamate receptors. So what we see is an early wave of increased chloride, which is due to this flux of salt water through the, mediated by glutamate receptor activation. But that goes away in a few minutes. It doesn't cause that much apoptosis. It's real, it's there, but it's transient, at least in this particular model. It might be in different situations, depending on where you are next to a capillary, away from a capillary versus infarcted tissue, there could be a bigger problem. But it isn't a problem that the cells can't overcome. What happens is in about an hour, over the course of an hour, we see the chloride start to go up again. So in the 5 minutes, 10 minutes, we see this glutamate-mediated peak. We know it's glutamate-mediated because if we put on glutamate antagonists, it goes away. But then there's this later peak that we can't block with antagonists. And that's that later peak of chloride and activation of caspases is the thing that we can block with metalloproteinase inhibitors. Now, one thing is, in terms of complexity, is we haven't proven that the metalloproteinase inhibitors are only acting in this sort of Donnan way, in an osmotic way. There are lots of enzymes that are anchored to the matrix and serve to tell the neuron, what is its tonicity relative to the extracellular space. And a lot of those are G protein linked enzymes that activate the transporters or change transport velocity and all kinds of other things. So that's how cells initiate, regulatory volume increases and decreases. So it is certainly possible that when we damage the matrix, not only are we sort of exposing the cell to the environment osmotically, but we're also disrupting a lot of its sensors that it uses to say what's going on in the extracellular space. And that might be, for instance, what triggers I don't really know what triggers the caspase activation, but rupture of those connections might be what triggers the caspase activation so reliably.
Thank you so much.
So for that, like the chloride homeostasis, I think you mentioned like the age difference, like in the adult, you use for neonatal, so it's within 24.
Right.
So what is that related with those? Is that like a protein expression difference of the?
Yeah. So it's a complicated thing, and I only got into part of it. But so in 2005, it seemed obvious, or in the early 2000s, probably up to 2014, that there was a transporter expressed early, NKCC1. There was a chloride accumulator, and a transporter expressed later in development that exported chloride. And as synaptic connectivity increases, you get more and more of the chloride exporter, which you need for GABA inhibition, and less of this chloride accumulator. But the other thing that happens is that as cells, neurons mature, they accumulate more and more cytoskeletal elements. So, you can't really make, a dendrite is a pretty amazing engineering feat. Axons even more amazing. They have these long, skinny processes. They have to have a very complex cytoskeleton to make that work. And so the concentration of proteins and nucleic acids go up in the cell over time. And so coincident with the switch from this watery cytoplasm to this very proteinaceous gel that's characteristic of adult neurons, you have this switch from a chloride importer to a chloride exporter. But both of the transporters can run in either direction. And the thing that we have a J. Physiol paper that's been cited, I think a total of 20 times or something, maybe even less than that, where we went through the sort of biophysics of it. But both transporters are reversible. So it doesn't really matter that much which one you have. The transporters are obligate co-transporters of salt and water. So the osmotic tension changes the reversal, but sort of the equilibrium point of both transporters. So you can have NKCC1 expressed in a cell that has relatively low chloride, but that changes the osmotic gradient across that membrane. And so the water can be tilted one way. You can have one water gradient that's like this and a chloride gradient that's like this, and those balance each other because neurons don't have any aquaporins. They don't have any a way for the water to sneak in and break up that balance. So it's a pretty complicated system, and it's unfortunately much more complicated than the yang and yin thing of NKCC1 and KCC2. But I think the advantage of this, of thinking about the Donnan effects, is that it explains so much more of the things that we see after brain injury. And so that's why we're more focused. Was that the thrust of your question? Okay, thanks.